From today, we are starting a new chapter about experimental designs. So what is meant by experimental designs, right? So what is meant by design and analysis of experiments? So we see the definition of design and analysis of experiments is planning an experiment to obtain appropriate data and drawing inference out of the data with respect to any problem under investigation. Right? So this is the definition of design and analysis of experiments. So in this definition, you can see that this has got different components. So let's see these components one by one. So the first component, of course, is any problem under investigation, right? So you have any scientific problem that you need to get the answer for, that you need to solve. So that is your problem under investigation, the problem that you are investigating, the problem that you're trying to solve. So what you do for that is you plan an experiment, right? So you have a problem under investigation. So what you do for that problem is that you plan an experiment to get the answer for that problem. So you plan an experiment to whenever you have a problem under investigation. So what is the purpose of planning an experiment is that as a result of your experiment, you obtain appropriate data, right? So the purpose of planning an experiment is to obtain a data which is appropriate. So now appropriate means that where this data is going to be used for. So the purpose of collecting this data or of obtaining this data is to draw inference out of the data, right? So these are the main components of your design and analysis of experiments, that whenever you have a scientific query, whenever you have a scientific problem under investigation, so what you're going to do is that you're going to plan an experiment to get the answer for your problem under investigation. And you plan an experiment in such a way that in the end of the experiment, you're going to obtain appropriate data. And you're going to use that data to draw inference, to draw a conclusion about the problem that you were investigating, that what is the solution to that problem, or what is the answer to that problem, to be more precise. So whenever you have a problem under investigation, you plan an experiment, you get your appropriate data, and you draw inferences out of the data. So these are the main components of design and analysis of experiments. So experimental design is basically a method to create a set of procedures for testing a hypothesis, right? So experimental designs, in other words, we can say that this is a method to create a set of procedures for testing a hypothesis. And again, we are going back to the definition that whenever you have a problem under investigation, so what you do is that you plan an experiment. So what you plan an experiment for, that you make an experimental hypothesis as an explanation to the problem that you are investigating. So whenever you have a problem under investigation, you make an experimental hypothesis to solve that problem, and then you plan an experiment to check that experimental hypothesis, right? So that experimental hypothesis is designed in a way that it can be checked through an experiment. So you plan your experiment to check your experimental hypothesis, and as a result, you're going to obtain some data from your experiment, and that data is then used by statistical analysis to draw inference about the problem that you were investigating, to draw inference that your experimental hypothesis, it stands accepted or rejected. So this is basically the concept of design and analysis of experiments. So we can basically further understand these components through an example. So the first component is any problem under investigation. So let's suppose the problem that we are going to investigate, or the question that we are going to answer, is that are there any effects of different fertilizer doses on crop yield, right? So this is the problem under investigation, to find out the effects of fertilizer dose on the yield of crop. So what you're going to do for that purpose is that you're going to make a hypothesis. So hypothesis could be that there is an effect of fertilizer dose on crop yield. So you plan an experiment for that purpose. So your experiment could be that you define doses of fertilizers to be checked, right? Because we're talking about the dose of fertilizer, the effect of the dose of fertilizer on crop yield. So for that purpose, what we are going to do is that we can plan an experiment in which we give plants, give crop plants different fertilizer doses. So we define the doses of these fertilizers to be given to the plants. So we can define 2, 3 or more categories of the fertilizer doses that we are going to give it to the plants. And the next part of your experiment is to grow crop plants receiving different doses of fertilizer that you have defined in planning your experiment, right? So you define different doses of fertilizers, and then you grow the crop plants and you give different plants different doses, and you see the effects. And then in planning of your experiment, you also have to define statistical analysis to be done. So defining the statistical procedure that you have to employ, that is a part of planning your experiment. So this is a necessary part of your experiment, that you not only design the physical infrastructure for your experiment, you also design or you also define the statistical procedures you are going to employ. So you have to define the statistical analysis that you can apply on the data that you obtain out of that experiment, right? So this is how you plan your experiment to obtain appropriate data. So what kind of the data are we going to get out of this experiment? So this data is, of course, crop yield and the different doses of fertilizer, right? So we defined different doses of fertilizers, we gave these different doses to crop plants, and in the end we are going to get the data. And this data is basically the crop yield and the different doses of fertilizer. So this data is obtained. So what is the next step, or what is the next component of experimental designs, is to draw inference out of the data. So what kind of inference are you going to have if different doses of fertilizer affect crop yield or no, right? So this is an example by which we can understand the experimental designs concept and the different components of experimental designs. That you have a problem under investigation. For example, you have to find out the effects of fertilizer dose on crop yield. So you're going to plan an experiment to get the answer for this problem. And in the planning of your experiment, you're going to define the doses of fertilizers to be given to the crop plants, and then you grow the crop plants under different doses of fertilizers. So they are going to receive different treatments, different doses of the fertilizer, and you also define the statistical analysis that you want to perform on the data that you obtain. So the data you're going to obtain out of this experiment is your crop yield. So crop yield of plants, or crop yield under different doses of fertilizer. So this is the data that you have. Then you apply the statistical analysis on this data and you get your inference. And you get your inference that if there is an effect of different fertilizer doses on the crop yield or no. So this is how you plan experiments and you get your results or your answers. So the experiments are different from observational studies, right? So what is an experiment? So in an experiment, you have to apply a treatment deliberately on a group of objects or subjects that you're investigating, right? So in case of experiment, what you do is that you define different treatments and you apply those treatments onto objects or subjects. You deliberately apply those treatments. So why do you apply these treatments for? To see a response. So the purpose of applying these treatments is to see a response, to observe the response of these treatments, right? So what you do is that you have your group of objects or subjects on which you're experimenting. You deliberately apply a treatment on those subjects, and then you see the response. That what is the response to the specific treatment that you had applied. While in case of observational study, what you do is you collect and analyze the data without altering existing conditions. So in this case, you're not applying any treatment, right? So you don't define a treatment, you don't apply a specific treatment to see the response of that treatment. Rather, you collect the data which is present as such, and you do the analysis on that data. So there is a difference between the experiments and the observational studies. In an experiment, you define a treatment, you deliberately apply the treatment onto your group of objects and subjects to see the response to that treatment. While in case of observational studies, you are not going to apply any treatment. Rather, you are observing, you collect the data from the existing conditions and you analyze that data to get your answers. So this is the difference between experimental and observational studies. We can further understand this difference between experimental and observational studies is by taking a couple of examples. For example, you want to see the effect of fertilizer dose on the crop yield, as we were discussing in the previous slide, that we want to see the experiment of to see the effects of different fertilizer doses on the yield of crop. So for that purpose, you plan an experiment. You define treatments, and the treatments are different fertilizer doses, and then you apply those treatments onto your objects, which are crop plants in that case. So you apply those treatments onto those objects and you see the response. The response is crop yield. So you get your response and then you analyze that response to see that what is the outcome of your experiment, and you draw inference out of the experiment. The observational study could be, if you take example from agriculture side, that you want to see the crop yield, the differences in the crop yields from different types of the soils. So for that purpose, what you're going to do is that you're going to collect the data of crop yield from the crops that have been grown into different soils. So you're not applying any kind of a treatment, you are just taking the observations at random, right? So you're just taking the observations, you're collecting that observation, you're not applying any treatment, and then you're seeing that is there any effect of different types of the soils on the crop yield. So you're not growing your plants under lab conditions, under your greenhouse, to see the effects of different soil types. You just go into the fields, you just randomly go into the fields and collect the data and analyze the data to see the response. So this is the difference between experimental and observational study, that in experiment you define a treatment, you apply a treatment and see the response. In observational study, you don't define a treatment, you don't apply a treatment, rather the existing conditions which are present, you take the data from those existing conditions and just analyze that data. So this is how the experimental studies are different from observational studies. Now let's discuss some concepts related to experimental designs. So we are done with the definition of experimental designs, and we have also made the differentiation between an experiment and an observational study. So now it is time to define some further concepts in experimental designs. So one of the concepts is the concept of testable hypothesis. So a testable hypothesis is a hypothesis that can be tested through an experiment. For example, in the case of the example that we are following about the crop yield and the effects of fertilizer dose on the crop yield, so the hypothesis that can be formulated in that case is a crop yield is affected by fertilizer dose. So this is the experimental hypothesis, and remember, this is not the statistical hypothesis, this is the experimental hypothesis that we have proposed. And this experimental hypothesis is a kind of hypothesis which can be tested. So this is a testable hypothesis. So this hypothesis can be tested through an experiment. And then we have the concept of independent variables. So one or more variables that are precisely manipulated. So the independent variables in experimental designs are those variables. Variables again means that they vary and that they have a variation. So these are the variables that can be precisely manipulated. So you can manipulate those variables. So these are known as the independent variables. For example, the amount of fertilizer or fertilizer dose. So 3 doses or 4 doses can be given. So this is an independent variable because the amount of fertilizer is not dependent on anything. It is not dependent on crop yield, right? So this is an independent variable, and this variable can be precisely manipulated, that you can precisely define the doses of fertilizer that you're going to apply onto the crop plants. So this is what is meant by independent variable in experimental design, that this is a variable that can be precisely manipulated. So this is basically the treatment that you're going to apply. And the example we are following, in that example, the independent variable is the amount of fertilizer or the fertilizer dose. So we can define that variable, we can define 3, 4 or more doses of fertilizer that can be applied on the plants to see the response. So the next type of the variable that we have in experimental designs is the dependent variable, and this is usually just one dependent variable in experimental designs. The independent variables could be more than one, but the dependent variable is usually only one in experimental designs. So there is one dependent variable that can be precisely measured, right? So the characteristic of dependent variable should be such that it is not something which is qualitative, or it is not something which is ambiguous. Rather, this is something which is quite obvious and which can be precisely measured. So this is the dependent variable. In this case, the dependent variable is crop yield, and crop yield can be precisely measured on scale. So a testable hypothesis is a hypothesis that can be tested. An independent variable is the variables that can be precisely manipulated, and it can be one or more than one variables. And the dependent variable, which is usually only one, and this is the variable that is basically your response variable, and this is a variable that can be precisely measured. And then we saw the examples with reference to the crop yields and fertilizer dose in defining these concepts. So then we have another concept, and that is the concept of experimental unit. So an experimental unit is a unit which receives a treatment, right? So a unit, an object, or a subject which receives a treatment is known as the experimental unit. A leaf, a tree, or an individual that receives a treatment is an experimental unit. And sometimes this is referred to as a plot, right? Especially when you're designing experiments for the agriculture field, then you sometimes refer these experimental units as plots. So in the example that we are following, the experimental unit could be a single crop plant receiving a specific fertilizer dose. Now remember one thing, that we are not talking about the group of plants, we are not talking about the group of objects, we are talking about a single experimental unit, right? A single experimental unit that is going to give us a single response, a single observation. So a single crop plant receiving a specific fertilizer dose is an experimental unit in this case. So if you have 100 crop plants in your experiment, that means you have 100 experimental units or plots, right? So you have 100 experimental units if you have 100 crop plants in your experiments. Now block, so what is meant by block is a collection of plots or experimental units. So a group or a collection of plots or experimental units is known as a block. And this is also known as samples or groups. So in the example that we are following, a group of crop plants receiving similar dose of fertilizer is known as a block. So in this case, what we are talking about is, we are not talking about the individual units, rather we are talking about the collection of the sampling units or experimental units. So a block is basically that collection or that group of the experimental units that are receiving a similar treatment or a similar dose of fertilizer. So in this case, a group of crop plants that receive similar dose of fertilizer. And of course, because we are giving different doses of fertilizers, so there are going to be more than one block in our experiment, right? So multiple samples, 2, 3, or more samples in an experiment. And these are defined by the group of the plants that are receiving similar fertilizer dose in this example. So what is an independent variable? It is the variable of our interest, and this is our treatment that we want to see the effect of. And what is our dependent variable? Our dependent variable is our response variable, and we are going to check our hypothesis through that response variable. So an experimental unit is that single object or that single subject which is going to receive a single type of the treatment, right? So a collection of the blocks, a collection of the experimental units that receive a similar type of treatment is known as a block or a group or samples. And because we are applying different doses of the fertilizer, or we are applying different treatments of our independent variable, so therefore we are going to have more than one block in our experiment. Now the sources of variation. The observations, they vary considerably among different experimental units, and this is known as variation, right? So the different experimental units that are going to give us different observations. So there is going to be a variation in the response in different experimental units. For example, different plants will grow at different rates and crop yield of the samples or blocks will vary, right? So we are growing the crop plants, in the example we are following, so we are growing the crop plants under different doses of fertilizer, and a single block will have a different value from the other one, right? So different plants, they are going to grow at different rates, and the crop yield of the samples, which depends on the growth of the crop plants, so that is going to vary among different blocks. So we are going to have a variation in response, we are going to have a variation in our observations. So what are the sources of this variation, right? So the two main sources of variations in simple experiments like this one are treatment and extraneous factors that cause experimental error. So these are the two main sources of variation that we are going to observe in our experiment, right? So what is meant by the concept of sources of variation? The concept is that our experimental units, they are going to give us different observations. The response of different experimental units is going to be different, and this variation is because of two main factors: the treatments, which we deliberately introduced, and the extraneous factors, which result into experimental error. Now let's talk about the sources of variation independently. So the first source of variation is the treatments, right? So treatment is a source of variation, and this is deliberately introduced. So this is done by manipulation of a certain variable of interest, and this is known as treatment variable, or the predictor variable, or independent variable, or factor, right? So these are different names for this independent variable that we are going to use, and the different treatments of the independent variable that we are going to apply on different experimental units. So this is one source of variation, and this is a source of variation in which we are actually interested, because we want to see the response, that is there any change, is there any difference in the response of different experimental units to different treatments. So this is very much the source of variation of our choice. So this is the variation introduced deliberately by the experimenter to see the influence or effect. This is our variable of interest, and in this example, this is fertilizer dose. So fertilizer dose is basically our variable of interest, and the treatments or categories of variable are different doses defined by experimenter, right? So the treatments or categories of this variable, they are different doses which are defined by experimenter. Now the other source of variation is the extraneous factors. Now extraneous means some external factors. So these are those factors that are largely uncontrolled, like the genetic variation among plants, the environmental factors that are affecting plant growth, the observer's fatigue, because if it is going to take measurements on the same day, then obviously this is going to result in an observer's fatigue. And the instrument effects. Of course, if we are taking observations, we are taking measurements from instruments, and there could be some error in the instrument that largely goes unnoticed. So that could result into a source of variation. So these are the sources of variation that are largely uncontrolled. These are known as extraneous factors. And these are unavoidable, and they result in a source of variation which is known as experimental error. So this is basically a variation produced by a set of known factors beyond the control of experimenter, right? So this is a variation that is produced by a set of known factors. So we know about these factors. We know about the genetic variation, we know about the environmental factors, we know about the observer's fatigue, and we know that our instruments, they can give an error. Sometimes they can give an error in the observations. So these are the examples of those sources of variation that we know and we cannot control. So these are unavoidable, and that they result into experimental error, and that is one source of variation. For example, in this case, the genetic makeup of plants and other environmental factors that affect the plant growth. So these are the examples of the extraneous factors that can result into experimental error, which is a source of variation among the response of different experimental units. And now we are going to discuss the principles of experimentation. So of course, when we are doing an experiment, we have to follow certain principles of the experimentation. Almost all experiments, they involve three factors: randomization, replication, and local control. So these three principles, or these three factors of the experimentation, they are in a way complementary to each other, and they complement each other to increase the accuracy of experiment and to provide a valid test of significance. So randomization, replication, and local control, they complement each other, and they complement each other to increase the accuracy of the experiment and to provide a valid test of significance. And they also retain the distinctive features of their own roles as well, right? So their roles are complementary to each other, as well as they have their distinctive roles as well. So these are the three basic principles of experimentation, and now we are going to see these principles one by one. So the first one is randomization, and as its name indicates, is that this principle makes sure that every experimental unit will have the same chance of receiving any treatment. So you're not nominating any of the experimental units that they are going to receive this specific treatment, or this group of the experimental units they are going to receive the treatment number 2, and this group of experimental units is going to receive treatment number 3, and so on, right? So there should be no bias in that, and the experimental units, they will have an equal chance to receive any of the treatments of the independent variable, right? So this is what is meant by randomization. For example, you have 100 units, right? 100 units or 100 plants. So every unit has equal chance of receiving any of the treatments, right? So you're not going to say that the first 10 plants, they're going to receive treatment A, the next 10 plants are going to receive treatment B or treatment number 2, or so on. No, this is not the case. You have 100 units, and every unit has got equal chance of receiving any of the treatments. So this is known as randomization, and this is a very, very important and basic principle of experimental designs. Now the next principle of experimental designs is replication, right? So replication means repetition. So repetition of experiment under identical conditions, right? Repetition of experiment under identical conditions is known as replication. So which means that if you're going to give treatment, so that treatment is to be replicated. So number of experimental units under the same treatment. So this is not enough in most of the experiments that you give treatment to only one of the units and you see the response, right? So that is not going to be sufficient. You should give that treatment to a number of units so that you can see the variable responses. So there should be a replication in your treatments, and greater the number of replicates, greater is the precision of the experiment, right? Because your treatment is not the only source of variation that you have in your experiment. As we saw, that there are different sources of variation that are largely uncontrolled, right? And they result into experimental error. So if you're going to apply a single treatment on a single unit, and you're deciding on that single unit for each of the treatment, like for you have 5 treatments and you have 4 treatments, you select only 4 plants and you want to see the effects of those treatments on those plants. So is that going to be sufficient? No, that is not going to be a sufficient response, because there are other factors that could affect the growth of the plant in that case. So you need to have more number of the sample units that are going to receive a specific treatment. So you're going to replicate your treatment on a number of sampling units so that you can increase the precision of your experiment. For example, you have 100 plants or units, and you have 4 treatments, which are 4 different doses of fertilizers. So what you're going to do is, are you going to select only 4 of those 100 plants and apply those 4 treatments, get those 4 observations, and draw your inference? No. You're going to replicate those 4 treatments on these 100 plants or units. So if we distribute them equally, then there should be 25 replicates of each treatment. So these are the 100 units. We have So these 100 units, they are going to receive different treatments, and these treatments are only 4. So these treatments are going to be replicated on different experimental units. So if these are 100 and we have 4 treatments, then every treatment is going to be replicated 25 times. And this is a good number of replication, and you can get a precise result out of this replication. Then the next very important principle of experimental designs is the local control. So control of all factors except the ones about which we are investigating. So local control is basically about those sources of variation that are unwanted, right? That are nuisance factors that are unwanted, but we cannot control them fully. But we should try to maximally control those factors, and we are not going to control the factors of or the sources of variation that we are interested in. For example, the treatments, right? So treatments are the sources of variation in which we are actually interested, so we are not going to control those treatments. We are going to apply local control on those sources of variation which are nuisance, which are unwanted, and which are unavoidable. So we are going to we are going to fully control those sources of variation, and if we cannot fully control those sources of variation, we try to minimize their effects. So the purpose of local control is to reduce variations due to extraneous factors. So the advantage of local control is that it reduces the variations due to extraneous factors that cause the experimental error. So we are going to have a little amount of the experimental error, and this increases the precision of experiment, right? So this also increases your precision of the experiment because you have less variation because of the experimental error, and more variation because of the factor of your interest, which are your treatments. For example, in the croplands yield example we are following, we can control the genetic variation, which is a source of experimental error. So we can get plant seeds from the same source. So this is how it can be avoided. This is how we can apply the local control, that we can get the plant seeds from the same source, from the same cross, right? From the same cross of plants, so that there is least genetic variation among these different plant seeds. We are not going to eliminate genetic variation because even in a single progeny, there is genetic variation, right? But we can minimize that if we take that from the single source. And then we can apply homogeneous environmental conditions that affect the plant growth, because this is not just the nutrients in the soil and we are going to mend those nutrients by applying fertilizers. So these are not just the nutrients in the plants that affect the plant growth. These are other environmental factors as well, like sunlight and moisture, and so these are just some of the factors that also affect plant growth. So how we can control the effect of these environmental factors, or how we can minimize the effects of these environmental factors, is that if we provide all of the plants or all of the units with the homogeneous condition, right? So they all receive the same amount of sunlight, they all receive the same amount of water, and they are kept under same temperature. So if they are applied with the homogeneous environmental conditions, then we can reduce, we cannot fully eliminate, we can reduce the variation which is imposed by these extraneous factors, and that results into an experimental error. So the purpose of local control is to reduce the experimental error to maximum extent. And this is done to make sure that the only effective source of variation are the treatments. So there are going to be sources of variation due to the extraneous factors, but those sources of variation are minimized so that they are not that much effective. So that we are left with only one effective source of variation, which is our treatments. So this is the basic purpose of local control. And another concept in the local controls is to have a control group. So it is important to include a control or a placebo group as well. So this is going to be a group of sample units that receive no treatment at all, right? So a sample or group of plants receiving no fertilizer, they are just grown in simple soil. So this is going to be your control group, and this control group also eliminates the also reduces the chances of experimental error. So these three principles of the experimental design: randomization, replication, and local control. So all of these, they're essential for making a valid estimate of error, which is within variance, right? So they all are essential in making a valid estimate of error and to provide us a valid test of significance, right? So we need to exercise these principles to maximum possible extent, so that we can have a valid estimate of error, so we can clearly identify that what is the source of variation other than our source of variation of interest, right? So what is the magnitude of that variation which is introduced by the experimental errors, and this is going to provide us a valid test of significance. It increases the accuracy and precision of your experiment. So if your plant, they are growing in different sunlight or under different temperatures, then you're not going to be sure that whether this is the temperature, or this is the sunlight, or this is the water content that has affected the plant growth, or is it only your treatments, right? So you need to make sure that your effective source of variation is the treatment, and you are also sure in calculating the variation which is introduced by the errors. So this should be exercised. And the attention to experimental design is important, right? Because the designing and execution of experiment directly affects the validity of the experiment. So again, taking the same example, that if you don't take care of the extraneous sources of variation, if you don't care about the enough replication of the experiment, then you're not going to increase the precision of your experiment. Then your experiment results, they're not going to be that much valid. So whenever you are going to do that experiment, you're not going to start it at random, that a thought comes into your mind, okay, I can check the experimental hypothesis by doing this experiment, and you start the experiment next day. No, you have to properly design the experiment, and you have to first take notes of the conditions that you have in which you can do that experiment, the lab conditions or the field conditions in which you are doing your experiment. So you need to take notice of all of those factors, and then on the basis of all that information, you are going to design your experiment, and then you are going to perfectly execute your experiment. And this is important because this directly affects the validity of your experiment. So the experimental designs, they are particularly important, and they need to be these principles of experimental designs, they need to be taken care of. Now, understanding research question and experimental conditions is extremely important, right? So this is the same thing that we are talking about, that you have to have a clear understanding of the research question and the experimental conditions. So clear understanding of research questions helps in deciding the levels of treatment variable, right? So why the clear understanding of research question is important? Because it helps you in deciding the levels of the treatment variable that you're going to apply on your units. And also the relationship between treatment and a response variable, you need to have a clear idea that if your treatment variable, what is the direct relationship between the treatment variable and the response variable? So is it a logical treatment to get that response or no, right? And a thorough knowledge of the confounding factors and field conditions is also important, as we discussed, that you need to have a clear understanding of the sources of variation other than your treatment. So you need to have a clear understanding and knowledge of the confounding factors and field conditions in which you are doing your experiment, so that you can apply a precise local control. And you also need to have enough replicates for statistical analysis. So understanding research question and experimental conditions is extremely important. Now, with this introduction to the basic concepts and principles in experimental design, we are now going to mention the basic types of the experimental designs that are used. So there are three basic types of the experimental designs that are used in biology, especially in the field research and the agricultural research. So there are three basic types of these experimental designs. The first one is completely randomized design, or CRD. So in this design, there is complete randomization of the treatment on experimental units. So this is the first type of experimental design. And the second type of experimental design is randomized complete block design. So in this case, there is also randomization, but this randomization is not exercised onto the whole whole of the sampling units. Rather, this is exercised within the blocks of those experimental units. We are going to see this detail, and then we are going to discuss this RCBD in detail. The third type of the experimental design is the Latin square design, and it doesn't have randomization as such, and we are going to see the details in the respective lecture of Latin square design. So these are the three basic types of the experimental designs that are used in field research, in biology, and in medical sciences.